Hello, everyone. My name is Patricia Montes. On behalf of our local ASBNB chapter, I want to welcome you to this Fall 2020 virtual edition of BEARS. As many of you know, BEARS is normally held in person, either in CSHIP or in Scanlan in the JAC. But due to the circumstances, we are holding this special version online. Just some quick things to know. This is one of 3 sessions, like I said, showing student submitted research and general science interest videos. Let's see more people coming in. Feel free to browse in between the 3 rooms. I'm going to post the links once again once we get started with the first video. The links should be in the programs as well that was sent out by the STEM Center a couple of days ago. There will be a total of 6 videos that we will show in this room, each followed by a brief 2-minute Q&A session. So feel free to just unmute yourself and ask a question to the presenters. The presenters will be in this Zoom chat as well during their video. And then just lastly, I'm gonna send out a link, and it should be in the program as well, post-attendance form that we ask you to fill out after we get done with the program today. So let me start the first screen share of the video. The video that I will be playing first will be presented by Melanie Puente. And the title will be Accelerated Telomere Shortening in Response to Life Stress.
Hello everyone, my name is Melanie Puente, and today I will be talking about telomeres and what they have to do with stress. I know this year has been quite difficult for many, and I hope this presentation will serve as a sort of caution for those who have been stressing out a little too much this year. But before I get into that, I want to talk about the structure and function of telomeres. Telomeres are made up of repeated nucleotide sequences, and in the case of humans, this nucleotide sequence is TTA-GGG. Now, the end of telomeres are not blunt, which means that one of the strands will have a 3' overhang, and this 3' overhang will loop back and form a cap-like structure called a T-loop. So these telomeres are located at the end of chromosomes, and the main purpose of telomeres is to provide protection. They protect the ends of chromosomes from enzymatic degradation, DNA repair mechanisms, and recombination. Now, something really interesting about telomeres is that our telomere length serves as a marker for biological aging. So as we get older, eventually over the course of many years, our telomeres will start to get shorter and shorter. Okay, and this is not necessarily a bad thing. We all grow old eventually, but speeding up the process of aging may also speed up the process of getting age-related diseases. So with that in mind, what do telomeres have to do with stress? Well, several studies show that too much stress leads to the shortening of telomeres. One study called Accelerated Telomere Shortening in Response to Life Stress had 2 groups of 58 healthy premenopausal women who were biological mothers of either a healthy child or a chronically ill child. They completed a standardized test of a 10-item questionnaire assessing their level of perceived stress over the course of a month. In the study, they then compared mothers who scored the highest and lowest quartiles of their perceived stress in the questionnaire and found that, independent of their chronological age and BMI, the high-stress group demonstrated significantly shorter telomeres. So how would stress cause telomere length to decrease? Well, in this particular study, they found that those who had shorter telomeres also had a higher oxidative stress. When oxidative stress increases, this also increases DNA damage and inflammation, and these factors in turn cause telomere shortening. Now, I think it wouldn't be fair of me to simply just say, hey, just relax. So I actually wanted to find some solutions and suggest a tip, which is exercising. I don't think it's surprising to hear that there's many benefits to exercising. And in this study, consistent with the previous study that I showed earlier, it shows that a higher perceived psychological stress is associated with shorter telomere length. And in addition to this, they also found that physical activity was able to protect those experiencing stress by buffering its relationship with telomere shortening. And furthermore, these findings support the CDC's recommendations for the amount of physical activity required to sustain a healthy body, which is 75 minutes of vigorous exercise per week. Again, to reemphasize the big picture here, what may come with aging faster is an earlier onset of age-related diseases, which means as opposed to having a higher risk for acquiring age-related diseases such as cardiovascular disease, cancer, and osteoporosis around your 70s and 80s, you may get them as early as your 50s and 60s. So what I want you guys to take away from this presentation is that even though we all grow old one day, there is something to be said about living a stress-free life. Thank you for listening to my presentation.
Good job, Melanie. Thank you for presenting today.
Thank you.
Questions? I actually would like to ask a question myself. Melanie, could you elaborate a bit more on what oxidative stress is and where it is found in our body?
Yeah, of course. So oxidative stress actually comes from the production of reactive oxygen species, and these species are usually produced in the mitochondria, so that's where you would find them. And so during processes such as cellular respiration, it's possible to accumulate oxidative stress.
Thank you. Very informative. All right, so yes, go ahead, Sarah.
If the telomere is destroyed, what happens to the chromosome?
If it's destroyed, the purpose of the telomeres is to protect basically the DNA, the chromosome. And so if the telomeres are damaged, that means that basically the chromosome won't be stable, and that's going to affect your DNA and cause DNA damage.
Thank you.
No problem.
I also have a question just to clarify. I think you said that oxidative stress leads to DNA damage and inflammation. Is that correct?
Yes, it increases.
Okay, so you said the shortening of telomeres can lead to oxidative stress as well? Is that what you said?
No, what I was saying is that oxidative stress causes it causes what did I say? It causes inflammation and it causes DNA damage. And then those two factors cause telomere shortening.
Okay, thank you.
Mm-hmm.
So my question was about I cannot pronounce anything. Oh my God, telomeres and stress, right? That what you were talking about? So
yeah.
What was, I didn't get to hear the beginning. What was telomeres? What was that?
What are telomeres?
Yeah, what is that?
Telomeres are found at the ends of chromosomes, and they basically they protect the chromosome from DNA damage.
Oh, and does that have anything to do with stress?
Yeah, so basically there are several studies that show that stress can actually lead to the shortening of telomeres, and usually that's naturally occurs through age, but they're saying that stress can actually accelerate this. So yeah, you should try to not be so stressed out, I guess.
Okay, thank you.
Mm-hmm.
All right, thank you guys for your questions. I think that we're gonna go ahead and start with the second presentation, which will be presented by Catherine Martinez. And her presentation is over a general science interest in bats.
Hello, my name is Catherine Martinez, and the topic that I wanted to use from class are keystone species. So I wanted to help explain this topic by bringing up an animal that doesn't usually come to mind when thinking about keystone species. So I want to begin this by defining what keystone species are. They're an organism that helps maintain biodiversity within an ecosystem, and they also help other species flourish and stop them from going extinct. One well-known keystone species is the bee, and they're a large pollinator that helps pollinate thousands of flowers and other plants. So the animal I wanted to bring up today are bats. Whenever you think of bats, good thoughts don't usually follow. You normally think of Dracula or vampire bats, and bats have always been seen in a negative way, yet they're very vital species to our ecosystems. Here's some reasons why we shouldn't fear bats and why we shouldn't really think of bats as a major part of the bat like family. So bats are actually really small. Vampire bats are actually really small compared to humans. The average vampire bat the length is 3.1 inches, and so that's very small whenever compared to human. And they also don't feed on human blood. They prey on larger mammals, for example, cows. Also, there are only 3 known species of vampire bats out of the thousands of bats. So whenever we think of bats, we really shouldn't think of just vampire bats. There are many other species like the Honduran white bat, the fruit bat, and many others. So now that we know why we shouldn't fear them and why vampires are a major role in the bat family, bats are a really unique mammal. They have social structures within their species. Like, for example, vampire bats, whenever one bat can't find any blood, they tap the shoulder of another bat and they will feed them. They're also the only mammal that can fly. So whenever you hear the name flying squirrel, they don't really fly, they actually glide. So the species I wanted to talk about today are flying foxes, also known as fruit bats. They tend to populate Southeast Asia, for example, Indonesia, and so they rely a lot on mangroves. And these bats are called flying foxes because they have very dog-like features. It's really cute. They have really big eyes, they have really big snout, and unlike other bats, they don't use echolocation. They have a really strong sense of smell and good eyesight. So these bats are nectarivores, I think that's how you say it, where they mostly feed on nectar. And one flower that this bat feeds on are the durian flowers, and this flower gives off durian fruit. So this durian fruit is a very popular fruit in Asia, and it's also known as the stinky fruit. Many Asian countries rely on this fruit for its economy, and when the flying fox is feeding on the fruit's nectar, or the flower's nectar, the bat is very fuzzy, so it collects a lot of pollen as it's feeding on its nectar, and once it goes from flower to flower, it pollinates every single flower. These bats are very gentle whenever getting nectar, unlike birds. These birds, whenever feeding on the flower, they tend to poke holes in the stem and destroy the flower. Flying foxes also help marine life, believe it or not. As I said, they rely a lot on mangroves, these bats, and so around these mangroves there's a lot of coral forests, and many fishes use these forests as nurseries. So as the bats are flying over the water, they drop feces down, and these feces contain nitrogen and carbon, and once they're released into the water, they make the organisms that live in that water a lot healthier. They make the coral healthier, leading to more homes for the fish. So these bats are very, very important to our ecosystems, and we really shouldn't see them in a negative way. Many bats are going extinct because of how we see them. So thank you for listening to my video.
And now I'll open the floor again for questions. If anyone has any questions.
Yeah, so like with coronavirus happening it came from bats. So I'm kind of wondering, how do you tie that in? Like, why did the COVID not strike the bat, but it affected us in such a big degree?
So like bats have like they've like adapted to all these viruses, and they like just hold them in, and some they don't they scientists believe that like the bats brought coronavirus, but they don't know how it became like contacted with humans. Some like cultures they believe that eating like a liver bat would bring you like good health, and instead that just like makes you like sick. Also, bats like have viral shedding due to like stress of being hunted down by like farmers and people who just like want to enjoy them for food.
Oh, that's cool. I did not know they had viral shedding. Thanks.
Thank you, Sarah. Any other questions? Please feel free to unmute.
Yeah, I have a question. So where is that fruit found that you were talking about? The Asian fruit where the bats eat? I forget the name of it that you said.
Oh, the durian fruit. It can be found a lot in like Malaysia and Indonesia, and it's a big part of farming over there. So many people aren't really educated over there about how these bats pollinate the durian fruit. So they end up like killing them because they think they're destroying the flower itself.
It's really interesting. Thank you so much, Catherine, for presenting and being here with us today. We're going to go ahead and move on to our third presentation of the day. And this presentation will be over biofilms, presented by Sarah Naranjo.
Hello, I'm Sarah, and today we're going to be talking about biofilms. So what are biofilms? They are a collective of one or more types of microorganisms that can grow on many surfaces. And this can be shown in fossil evidence that biofilms have been dated out to be 3.25 billion years old, which is actually insane. So you can see these in hydrothermal rocks in Australia that are actually 3.2 billion years old, and hydrothermal environments such as hot springs and deep sea vents, which leads to the question of, were they the reason where we are today? Are they the origin of life? So let's go more in depth. So the gooey substance that's seen in biofilms is called the extracellular polymeric substance, the EPS. And this is a network of sugars, proteins, and nucleic acid. And this is critical for the structure and the survival of biofilms. So without this structure, they can be destroyed by UV light, harmful substances such as bleach and toxic or contaminants of metals. So this EPS is this protective barrier in order to protect them from these harmful substances. And that's why it is so critical to understand why biofilms can be very destructive in the human body, because antibiotics that go into it need to have the cellular activity, and bacteria can be dormant in biofilms. So the antibiotics are of no use. So it's very important to understand where biofilms come from and to protect yourself from them. So let's look at the biofilm formation. So you first start off with this free-floating bacteria that attaches to a surface, and then this surface starts to secrete this EPS, which is this gooey structure. And then once that starts to begin, it starts developing the biofilm, and then when they mature, and then this bacteria is released and then put onto a different surface. That's why it's very easy to contract all these different things. So pause the video for now and then stop and think about the following questions. What are some examples of biofilms, and do biofilms have a negative or positive effect on the environment, and why are they so important? So really think about these questions and see what you can come up with. Okay, so the most common example is the dental plate that's seen in your teeth. So if you don't brush your teeth a while or a course of time, you start developing this thick consistency between your gum and your tooth, which can lead to very disgusting things later on in the future and eventually have to remove your teeth. So make sure you brush your teeth. But as you can see in these pictures, you see this white substance that's growing on it. So you see this thick layer and it's like gooey, which is the EPS in the biofilm. This is a very common example where you can see biofilms. Another example is oropharyngeal candidiasis, which is on the tongue and is most common in babies. You can see this because of the immune deficiency that they have, and that's why they cause this. And you can see in vaginitis, where it's the inflammation of the vagina and which causes a yeast infection, which can happen from catheters or different things that already have the biofilms. So make sure that's why it is very important to have a sterile and very sterile cleaning medical devices. So then things like this won't happen. So it actually has both negative and positive effects on the environment. You can see negative effects by how it leads infections if grown on medical devices, as discussed before, like breast implants, prosthetic cardiac valve, pacemakers, and catheters, of course. And the bacteria within a biofilm is much more resistant to antibiotics because they can be dormant and how the antibiotics have no use. And it's positive in how it's can be used in bioremediation, how it can degrade harmful compounds, such as treating wastewater, heavy metal contaminants of chromate, explosives, and radioactive substances. It can also be used in microbial fuel cells in which bacteria actually converts to electricity, which is a very interesting topic, and it could be very beneficial in the future. So why it is important? Because it is hypothesized to be the origin of life. And we wouldn't be here today without them. So how the origin of life began was in the water, and within biofilms, there's a very common characteristic of all of them is that moisture is involved. So this could be a very, very good theory of how we became who we are today. So thank you.
Thank you so much, Sarah, for sharing that with us today. Very informative. I would like to open the floor up again for questions for Sarah, if anyone would like to ask something.
Oh, and by the way, at the end, it's not a theory, it's a hypothesis. Dr. Amin got on me for saying theory.
We're saying that, yeah.
Yeah, so just a clarification.
No worries.
I have a question. This is Dr. Roselle. Hi, Sarah. What got you, I missed the very, very beginning because I was in another room, but what got you thinking about biofilms?
So in lab, we actually touched the topic, and it was interesting seeing how it can just grow anywhere. You can see them growing on any surface, and how it kind of, even with antibiotics given to it, it really resists that. And like, you can find that in your body or in other animals, such as like it's just very common everywhere. So it's very important we know about it, because it can be very harmful in the future if we won't listen to it.
In your experiment that you did for the Bio1 lab, did you by any chance, were you able to see any biofilms?
So we did our experiment, our project was the bread mold.
Okay, good.
So we have like, well, my bread was the only one that grew mold, but it had a lot of different colonies on there. And it was just kind of surprising seeing how mold colonies can just be floating around in your room or your house. And you just put it on a bread and put it in a bag, and it just grows out of nowhere. So it's very interesting because it's really common and it's seen anywhere, and we don't really know about it that much because it's not really talked about that much. So
yeah, they're considered yicky.
Yeah.
Well, great presentation.
Thank you so much.
If there's no more questions, then I will move on to the next presentation. And the title of this presentation is methionine aminopeptidase inhibitors in mammalian cells.
Hi, my name is Jamila Bukhari, and my team and I have been working on determining the cytotoxicity of a potential HIV tuberculosis dual-acting drug therapy in order to combat the HIV tuberculosis co-infection. One of the most interesting reasons for our research is that tuberculosis is the leading cause of death in people with HIV, caused by the bacterium Mycobacterium tuberculosis. Tuberculosis acts synergistically with HIV to target a patient's immune system, which can lead to an HIV tuberculosis co-infection. If we take a look at figure 1 over here, we can see the treatment success rate for new and relapsed HIV-positive tuberculosis cases. Towards the right of figure 1, from 2012 to about 2017, we can see that the amount of not evaluated cases has gone down, which is in light gray, but we can see that the amount of people that have died from this co-infection has kind of consistently remained the same here in orange. What this indicates to us is that we've not really found a treatment for this co-infection, but we've just gotten better at identifying the disease. The reason for this is that treatment is difficult. Drugs and therapies that target one disease can lead to increased susceptibility to the other. And so our collaborators at Texas Southern University have found a potential dual-acting HIV tuberculosis drug therapy dubbed UST-001, which targets the enzyme methionine aminopeptidase. This enzyme works to cleave the N-terminal methionine residue of polypeptide chains. Deleting this enzyme is fatal, which makes the enzyme a suitable drug target for the study. Methionine aminopeptidase is also universally conserved, and we can see here in figure 2 an overlay of the Homo sapiens methionine aminopeptidase and the Mycobacterium methionine aminopeptidase, and so you can see that they're very similar. Therefore, in vitro studies must be performed to determine the cytotoxicity of inhibitors to this enzyme in mammalian cells as a preliminary step in the drug discovery process. We were using a spectrophotometric assay, which was called WST1, to test the mammalian side of toxicity of UST-001, but the data obtained from those assays were inconclusive. We are now using a more accurate flow cytometry method and the cell count and viability in these assays to find the LC50, which is the lethal concentration, which 50% of the cell population are affected. We hypothesized that for human lung carcinoma cell line, which is our H1299 cells, the LC50 will be greater than 25 micromolar, which is low enough to not affect the survival of Mycobacterium tuberculosis. So our methods include first washing and lifting our H1299 cells, and then we seed our 6-well plates. We incubate those plates for 48 hours at 37 degrees Celsius, and then we conduct a new cell count and viability assay. We conduct a triplet blue assay in addition in triplicate to confirm those used results. So if we take a look at the results in the top right, figure 3 actually shows our average percent viability against our UST-001 concentrations. There are 3 negative controls, media, INH, and DMSO, and our concentrations consist of 1 micromolar to 100 micromolar. Our 3 controls and 3 lowest drug concentrations, which are 1, 5, and 10 micromolar, were all found to be greater than about 80% average viability, which was not found to be statistically different. However, at 50 micromolar UST-001, the average percent viability of H1299 cells was found to be 63.4%, which is significantly different from the controls. In figure 4, we can see a population and viability profile of one of our 3 triplicates from that 50 micromolar UST-001 used data with 66.5% viability over here. So at UST-001 concentrations above 10 micromolar, we did see that there was minimal cell death of cancerous H1299 cells. The viability of H1299 cells was lowest at 63.4% at 50 micromolar. It did, however, increase at even higher concentrations of 75 and 100 micromolar, which is a problem that we are currently addressing. It is possible that at these high UST-001 concentrations, the inhibitor is somehow being exported or being degraded by the cancerous cells due to their higher mitotic rates and their large amount of repair pathways. We would like to see the cytotoxicity of UST-001 in healthy non-cancerous cells with our second cell line called MLG2908, which is a non-cancerous mouse lung fibroblast cell line. We did perform a preliminary trial with the MLG2908 cell line, but it showed inconclusive data and no significant difference from our controls. We also hope to use a Muse-Gannex and VA apoptosis assay in the future, which will focus on the UST-001 mechanism of cell death, figuring out the cytotoxicity of UST-001 in mammalian cells, and finding no significant difference in the MLG2908 cell line will help us determine if UST-001 is in fact a viable target for drug development. Thank you.
Any questions over Jamila's presentation? Or Jamila, would you like to explain a little further anything about your research program?
Yeah, so first I want to apologize. I didn't realize it would be this blurry. But basically what we're trying to do is, what's really difficult is that people with HIV are very susceptible to getting tuberculosis, and then when they have that co-infection, it's really hard for them to treat them. So drugs that usually are, used to help with the symptoms of HIV are, they usually conflict with drugs that help with tuberculosis. And so basically what we're trying to do is find that middle ground in order for patients to actually not be treated because, but kind of just like ease those symptoms. And so what we want to do is first find the LC50 and then move on to the mechanism of our drug and hopefully see see how that kind of influences the co-infection.
It's very interesting. Thank you so much. Any questions from anyone?
Yeah, I have a question. I was gonna ask, have you found any like drugs that like work to treat or no?
So what we're doing right now is we actually have a collaborator at TSU, Texas Southern University, that made our potential drug. That's what we're trying to test. So we just named it UST-001. And so once we finish testing this drug, we do have other potential drugs that we'll test, just depending on our success with this one. But yeah.
It's very interesting. Thank you so much, Jamila, for taking the time to present today. And we will move on to our next presentation, which is by Francisca Gutierrez. And her presentation will be over the relevance of cancer, melanoma, and stress.
Hello everyone, my name is Francisca Gutierrez and today I'm going to be talking about the relevance of cancer, melanoma, and stress. So here we have a diagram of the layers of the skin, and we have the epidermis layer at the top, the basement layer, which is thin in the middle, and then the dermis layer at the bottom, where the blood vessel is located. So in the basement thin layer, we actually have these cells that are called melanocytes that are in charge of protecting against UV rays from the sun and aid in skin pigmentation. So whenever UV rays damage portions of the DNA that are in charge of cell replication, these cells can undergo rapid division, cell division, creating a tumor, which is melanoma. So whenever this tumor actually makes contact with the blood vessel, it's gaining access to nutrients and oxygen, which help its progression, which is a process called angiogenesis. And by also reaching the blood vessel, it has access to now move throughout the blood vessel and into different parts of the body in order to invade that tissue, which is a process called metastasis. So why is cancer melanoma important? Well, right here we have two pie charts with the figures for the 2020 approximate diagnosis of melanoma along with the 2020 approximate deaths of melanoma. So we can see that you have a large amount of diagnosis of melanoma with perhaps not as large comparison to deaths of melanoma. However, melanoma still continues to be one of the majority of skin cancer deaths, despite the fact that it accounts for just 1% of the types of skin cancers. This is an ongoing issue whose rates are rapidly increasing over the past few decades due to the proficiency of cancer melanoma to be able to metastasize to different parts of the body. So what does this actually mean in regards to stress and the actual cancer melanoma? So when an individual is stressed, it actually triggers the central nervous system to release hormones that are called catecholamines. In this example, I'm using norepinephrine. So norepinephrine actually binds to the cancer cell and to the tumor-associated macrophage that is in red. So this tumor-associated macrophage actually is derived from a monocyte, which is supposed to be converted into a macrophage, which is to, help the immune system. But certain cancers actually change, the distinction of the monocyte into tumor-associated macrophage, which in turn, after having contact with the norepinephrine stressor hormone, they release certain cytokines, which are proteins that help in the process of angiogenesis, metastasis, and just the
proliferation in general of this type of cancer. So what is the holistic point of view that we're trying to accomplish here? So in a practical lens, this means that we're just trying to raise the awareness through this evidence that there is a negative correlation from stress that an individual encounters with the level of proliferation of the cancer. So it is the aim of studies like this to hopefully increase the implementation of therapies that increase a priority in the treatment of mental health and wellness of the individual that is currently undergoing the stressful situation of this cancer disease. Thank you.
Thank you for the presentation, Francisca, but I believe that she's not here today to present for the Q&A. So I will just move on to the next video, which will be our last video. So our last video for today will be presented by Jacqueline Henderson. And the title of the video is Genetic Mapping in C. elegans.
Hi everyone, I hope you're all doing well. My name is Jacqueline Henderson, and today I'm going to be presenting genetic mapping in C. elegans. And this is part of Dr. Alexandra Simmons' research. So what is genetic mapping? Genetic mapping can offer firm evidence that a disease transmitted from parent to child is linked to one or more genes. Mapping also provides clues about which chromosome contains the gene and precisely where that gene lies on the particular chromosome. Genetic maps have been used successfully to find the gene responsible for relatively rare single gene inherited disorders. To produce a genetic map, researchers usually collect blood or tissue samples from members of families in which a certain disease or trait is prevalent. Using various lab techniques, scientists isolate DNA from these samples and examine it for unique patterns that are seen only in family members who have the disease or trait. These characteristic patterns in the chemical bases that make up DNA are referred to as markers. These markers don't by themselves identify the gene responsible for the particular disease or trait, but they give us roughly an idea of where that gene is located on the chromosome. In my own research on C. elegans, we use SNPs, which are a particular type of marker. So right here, this is a video of the C. elegans exhibiting the tremblor mutation. This is our current research at St. Thomas. So we are using genetic mapping to determine where the tremblor mutation is located on the chromosome of C. elegans. So right here you can see it's a tremblor mutation, so they're exhibiting constant muscle spasms. A C. elegans is a small nematode worm that, when mutated, exhibits this muscle spasm movement. We suspect that this is due to a mutation in the nervous system, and in the summer of 2012 is when Dr. Simmons started this research. So we obtained a particular strain of C. elegans that exhibits this involuntary muscle spasm. They are a perfect model organism for us because they exhibit only one crossover event. So the goal of our project is to locate and sequence the mutation that is responsible for the presumed involuntary muscle spasms in the mutant strain. Once we identify this mutation, we will conduct further comparisons to find homologous human genes, as it may be possible that this mutation could be responsible for a disease or a particular phenotype in humans. This is a test subject to try out specific gene therapies, knockout or knock-in therapies, test drugs, etc. So these are all my references for this presentation, and I'm looking forward to hearing all of your questions and feedback. Thank you so much for listening.
Thank you so much, Jacqueline. Any questions for her?
What has been your favorite part about the research that you've been doing?
So I've only been in this research group for about a year now, and we actually just finished chromosome mapping, which is the first part to genetic mapping. So that was really cool to find out exactly which chromosome that the mutation was located on. But now we're just waiting to get more DNA so that we can find where on the third chromosome that it's located. So I really liked chromosome mapping. That was really fun.
Where's the DNA coming from? Like, what type of DNA are you guys getting?
So we work with the lab at Baylor, and they gave us a sample of the worms that have the mutation. So we just extract their DNA and we use that to perform like PCR and run their DNA on the gel.
Okay, that's super cool.
It's really interesting. I really like it.
Good luck on further research.
Thank you.
Thank you, Sarah, for the question. And Jacqueline actually had a small question for you as well. Do you think you could elaborate a little more on why C. elegans is a model organism for this research?
So they actually exhibit very similar genes to humans. So kind of the goal of finding out where the mutation is located is for research in human diseases.
Very interesting. Thank you.
Thank you.
Any last lingering questions from anyone?
Okay, I have a question for, her name was Melanie?
Yes.
Melanie. Oh, that name is so pretty.
Yes,
Okay.
I'm here.
And once again, thank you guys so much to the presenters and to the viewers asking your questions. I'm going to send the post attendance form one more time if you guys didn't catch it. It's also at the top of the program, just so we can get some feedback from you guys. And in the form, there is a section where you can rank the presentations because the top rankings will from each room will receive a prize. It's a really cool 3D printed bear for presenting here at Bears. So it's really cool. They look really cute. But yeah, thank you guys so much. I hope you guys have a nice day.